Welcome, Bonjour, to Wahsa Distance Education Center's radio Zoom classes. This is SVN3E, Grade 11 Workplace Science, and I am Bronwyn Slade. If you'd like to participate live today, you can call the Wahsa studio at 1-800-465-7144 or at 737-4017. You can listen on the radio at 91.9 FM and also on the television at Bell Express View Channel 972. You're always welcome to join me live through the Zoom link, which is available from me, your teacher, and also from your DEC. Our classes are scheduled from Monday through Thursday from 3 till 4 in the afternoon. And we are now starting our 8th week out of our 9-week course. Only 2 weeks left. A reminder that all of your work is due by the end of next week, by June 10th. This week is the last week that I will be teaching new material. Next week is a review week. So I'll go over all of the 7 units that we covered with 1 or 2 units a day next week. But this week I will be finishing up Unit 6 and completing Unit 7. But all of your work needs to be in by June 10th, which is the end of next week. So you have just under 2 weeks to get that done. A reminder that the key questions are what you submit for grading, and they're all listed at the end of each of your IL lessons. So they're not all of the questions in the workbook, but specific questions, though they are most of the questions in the workbook, but they're just specific questions that are listed at the end of the IL book. So make sure that you follow that. Some of them are check your understanding questions. There are a few activities throughout, and then the review questions of each chapter. Remember to explain your thoughts completely. So really don't just give me one word answers, but give me full thoughts, full answers, so that I can really understand that you understand and that you're actually answering the question that's being asked and not just topic on the topic. You can do this either by hand or electronically. Either way is totally fine for me. If you'd like to write in your workbook, you can. The spaces are small, so I don't really suggest it, but if that's what works best for you, go for it. Just make sure I can read it and that you're giving me full answers. If you want to do it electronically, Word and Google Doc files are the easiest ones for me to open. Everyone has access to Google Doc if you use your NNEC email address. So go ahead and do that if that is what works best for you. That's totally great. If either of those files are not the file types that you'd like to use, if you'd like to do something different, most likely it's fine. Just let me know so that we can make sure I can open your files, which is the most important thing. Okay, so there's 3 different ways to submit your work. The first is to scan your work in and send it electronically. So if you've written it by hand, you can scan using a smart device. If you have an iPhone, the Notes app works, and if you have an Android phone or device, the Google Drive app works. Both have fairly straightforward scan functions, which I can support you with if you'd like. If that doesn't work for you and you need to take a picture to send your work, that is fine too. Don't worry about it. Scanning just makes the files a little bit smaller and easier to send. But if it's not easier for you, don't worry about it. Then you can send it to me either through email at studentwork@nnec.owen.ca and CC it to bronwyn.slate@nnec.owen.ca, or you can send it to me through Facebook Messenger at @vslatewahsa. The second method is to drop your work off in Sioux Lookout. We have an outdoor mailbox at our location 74 Front Street. We're the bright red building next to the post office, and we have a small white mailbox next to our side entrance. So you can just put your work in there at any time, and I will get it and get it back to you. Then the third method is to hand your work into your DEC. So your DEC can either send your work through the Express or fax it to 807-737-1732, or toll-free fax to 1-800-463-7852. If you'd like to connect with me through social media, both my Facebook and my YouTube channel are under the name B Slate Wahsa. You can friend me on Facebook if you'd like, or subscribe to my YouTube channel, and then you'll get notifications every time that I upload one of our lessons. All of our radio Zoom classes are recorded, and I upload them to YouTube, usually within a day or two. Sometimes it takes a little longer. And they're all on a playlist called SVN3E. So that can be a really easy way to access all of our recordings, all of our lessons. It's all right there. Also, there's a playlist that's called Supplementary Videos, which has all of the videos that I've shared in class, links there, so you can find them from their original resources. Science is a really visual subject, so I strongly encourage you to access the videos. This is going to set you up for the most success. I do a lot of integration of videos, of graphics, of images, so that to really give you a full understanding of the concept. So being able to watch it is really going to give you the most information. If you can't join me live through Zoom or watch the YouTube channel, let me know and I can send you a copy of the videos on a USB drive or something like that so that you still can access them. Alright, so if you have any questions or concerns, please reach out to me. I am checking in with students at this point, but if I haven't heard from you in a while, or I've never heard from you and you are working away, please reach out and touch base with me so that I know that you're working on the course, and that gives me some preparedness so that you hand in all of your work. I know that it's coming and I can make sure that we don't kick you out of the class before you're actually done. Paperwork and things are starting to roll, so reach out to me in case I don't know that you are going to hand in work. My email address is bronwyn.slate@nnec.on.ca. My Facebook is bslaitewahsa. The phone of the office is 807-737-1488, and my extension is 2209. You could also call toll free at 1-800-667-3703. I'm here Monday through Friday, 9 till 4. Though I do teach the first hour and the last hour of my day, so that's not a good time to try to get hold of me. And then another reminder about June 10th, I'm going to keep saying this over the next 2 weeks as it's super, super important. You need to get all of your work in by June 10th. Also, if you are hoping to complete this course, not only do you need to complete all of the 23 lessons in your IL booklet, but also you need to do a culminating assignment. You need to do a final project. So at this point, I'm not likely to get all of your work back to you just because things are piling up. I may get some of it back to you before the end of the term, but I won't get it all back to you before you have to do your culminating assignment. But you really need, so getting it in as soon as you can means I can get it back to you as soon as I can, and gives you time to do your final project, which you have. It's a take-home project that's going to take you a few days to do. So it's super important to get your work in. I think it's important to position myself in the context of education and learning as a person with power. So I have white settler ancestry, I have white privilege, and this means that I have a certain amount of power that comes with that identity that I did not earn and that I do not necessarily realize all of the time. So I work to constantly disrupt these power cycles and to be aware of them. I don't have it figured out. I don't do it perfectly, probably very much of the time. I am trying to do better and trying to learn more and more. But that is part of how I have become a teacher and the ways that I have existed in educational spaces. So my perspective may be very different than yours, and I would love to hear what you have to offer so that I can learn more. I do live in Northwestern Ontario on the traditional territories of the Anishinaabe people, and this is a culture that I am attempting to learn from when I can, when it's appropriate for me to do so, and integrate those teachings that are appropriate for me to integrate within our course in order to expand the horizons of what science is and what science education is, outside of just a westernized, Eurocentric perspective of what it means to be a scientist, what it means to use and interact with the natural world. This is my first time teaching this course, so it has been a very big learning experience, and I continue to have more things to learn and unlearn. It is a process, and I can recognize that as we're getting close to the end, that there's still lots I can do to improve. So if you have any feedback, feel free to share that with me. Also, our textbook is really Eurocentric. I really noticed that the way that it positions scientists and communities are focused very much on the white experience, and that's highly problematic and embraces a lot of people's experience and knowledge. So particularly, I've noticed it erases and ignores Indigenous Inuit and Métis both their knowledges and their experiences, and this is highly concerning and quite problematic. So I have tried to work in some examples and some knowledges that I do know. It is a work in progress, and I hope to do better in future years. If you have any suggestions of how I could do better and how I could integrate more knowledge and experiences of Inuit, Indigenous, and Métis people, please let me know so that I can do better. All right, we continue to be in Unit 6. So this is healthy ecosystems, and just a reminder of how connected ecosystems are. The population of frogs and other amphibians has dropped since the 1980s. So it seems as though 170 species have now gone extinct, so are completely gone. This seems to be due to global warming and the climate crisis. So this means that now these ecosystems are being adjusted because tadpoles usually eat algae that use up dissolved oxygen needed by other aquatic organisms. So now that this algae can thrive and is now using up that oxygen, which means that other aquatic organisms are not able to use that oxygen. Less frogs eating mosquitoes, flies, and insect larvae means that there are more mosquitoes, flies, and insects. Also, less frogs means that other predators that eat the frogs don't have that source of food, and that means that their numbers are dropping, which then will continue to have a ripple effect out on those ecosystems. So ecosystems are, in order to be healthy, need all of its members. So right down to those mosquitoes and flies, they have a role to play within the system. That means everyone is able to thrive together. So if one of them is threatened or wiped out, it affects the others, and it makes everything endangered. So we have talked about different species and how we identify them or how they're different and work within an ecosystem. Today, in lesson 20, we are going to talk about how we measure and monitor those species and their biodiversity, so that we can continue to learn and make sure things are doing all right for various species. So our learning goal is that at the end of this lesson, you will be able to describe common methods of measuring and monitoring biodiversity in various habitats. So you know you've done this because you can explain the differences between quadrat, line, transect, and aerial photography sampling. You can explain and calculate the Lincoln index, and you can describe the alphanumeric wildlife radio tracking and GPS methods of tagging and tracking. So, according to your workbook, which was published in 2010, so that's 12 years old, which isn't a huge amount of, it's not really, really old compared to some things that I'm working with, but it's a little bit old. Things are changing. Things are moving. In Canada, there have been 71,000 species of plants and animals that have been identified and listed, so recorded about. But we know that there are many more still undiscovered. So most of the larger organisms, like mammals, birds, and trees, have been almost completely identified. We can see them. They are easy to find and easy to track and see if they're different, and so we are easy to record information about them. But it's the smaller creatures that account for most of the unidentified species in Canada. For example, over 1/5 of all the species in Canada are insects, and we don't know what they all are. There are estimates of how many species of the more obscure groups, such as the nematodes, I don't know what that is, are a little more than guesses. We really don't know. We're sort of speculating based on other patterns, because we're not able to count them or track them in the same way. So here is a list of some of the larger groups of species. So again, this is not an extensive list at all, but you can see how fungi, we know of 11,800 different kinds of fungi, and there's an estimated 3,800 of fungi that we still don't know. We know over 3,800 flowering plants, and about 75 we think that we don't know. Coniferous plants, we know we have tracked 34, and we think that there's 0. We think that we know all the coniferous plants, which I would assume are trees. And then other plants, we know about 1,100, and there's about 60 they think that are unrecorded. So this continues with sponges, mollusks, arachnids, crustaceans, insects. See, insects are, we know 18,530, and we still think we don't know 11,800. So that's still a lot that we don't think we know. Fish, amphibians. We think that there's 2 amphibians that are unrecorded, which kind of seems ridiculous to me in terms of this list. It's like, if you know that there's 2 that you haven't recorded, then how come you haven't recorded them? But I don't quite understand how this list is generated. This is just sort of giving us a general idea. Then reptiles, birds, and mammals. We think that we know all of them, and that there's none that are left to be recorded in Canada. So how do we know that we know them or we don't know them? So in order to measure and monitor species biodiversity, researchers use different estimation methods. So they also use different technologies to measure species populations sizes, and that's what we're going to be looking at today, about how we're able to create lists like that when we're saying that we don't know all of them, or that we know how many of a certain amount of things are, which we didn't look at, but how do we even guess that when we can't count everything? So we're going to look at sampling, which is how we count things in science. So sampling is a way to measure and monitor biodiversity. So researchers monitor species biodiversity by collecting and analyzing repeated data and observations about the number of different species within a given area over a period of time. So doing this allows them to evaluate any long-term changes in the number of different species. So this is how tracking, counting, and collecting information about the various species, and then returning to the same space a period of time later, means that we can then see how things are changing and what is going on. Even within a geographically small habitat, it is difficult to go out and count each and every species that lives within it. Instead, researchers take samples of species within a habitat. Sampling is a way to give an estimate of the number of species in a given area instead of counting them all. So sampling means that by nature, we aren't assuming that we are going to exactly count everything. Everything is an estimation, but we're able to get a good approximation based on the information that we do count. So let's look at a couple of different types of sampling. So first we're going to look at quadrat sampling. So this is when a series of gridded plots are placed into a habitat. Then researchers identify, measure, count, and record data about the species within the plot. It can be placed randomly throughout the study area, or it can be in planned locations. The shape and size are uniform throughout a study. So if you're going to decide that you're going to do 1 meter squares, then you're going to continue to do that for the entire study so that you have a uniform measurement tool. But from study to study, your quadrats, could be square, they could be rectangle, they could be circular. They don't have to always be. It's not like there's one uniform measurement that is like, this is the only way to do quadrat sampling. No, you just want to be uniform within your own study. Your quadrats can vary in size, but often they're roughly 1 meter squared, just in terms of a general idea of how big they are. So not only are the different types of species counted and recorded, but the abundance is also measured by counting the number of species of each species found in a quadrat. So you know, oh, I picked a spot where there are dandelions are busting out. So that the fact that it's not just one dandelion, it's 100 dandelions is different than in a space where there's just one dandelion. So that is part of the information that's recorded. Here are 2 pictures of quadrat sampling. So you can see that they can be really different sizes. We have a small one here that looks like it could be like maybe 2 feet by 2 feet, and that they're looking just in a grassy sort of area and noting the plants in that area. And here is another one that is maybe, I don't know, maybe it's 1 meter by 1 meter, or maybe 1.5 meters by 1.5 meters. And then even within that, there are further little quadrats or little divisions inside of there to further analyze and get that information. about the species that are in that area. So it can be either passive or active sampling. So it depends on the organisms and the habitat being studied, which one you would choose. Generally, quadrats these first types of samplings are good for species that don't move. So either plants, lichens, mosses, things like that, but also like mollusks or barnacles or other animals that are stationary. But still, passive sampling is when none of the organisms or plants are removed. Everything stays, you're just looking. So researchers carefully sort through each quadrat by hand, or they can take photographs for future analysis. So they're not picking plants, they are just looking at them and noting them without really disturbing the area too much. Active sampling is when the organisms and plants found within the quadrats are collected for later identification. So this is often used when researchers are estimating the population of an organism within a study area. So that's just how you might choose which to do. So now we have line transect sampling. So this one, researchers lay down a long string or rope across a study area. The data is then collected either continuously along the line or at intervals along the line. So it can just be the whole line, or it can be every 5 meters or every 4 feet or something like that. It can be used on its own, or it can also be used in combination with a quadrat sampling. So you could have your line laid out, and then you could be using quadrats at those every certain intervals. Researchers record and count species over, under, and touching the line. They might also record species they don't see, but expected to see. Often when there are obvious geographical differences in the study area, the line might be laid out over a river or through a forest into a field. So if you're looking in an area where there are distinct differences, having that continuous line where you may, you'll be able to know where the changes are can be helpful. So here is a image. I wasn't able to find a great image. I wasn't super happy with this, but I guess it's hard to just like draw a line in the world. So here is a forest area, and then this is the transect line, and the researcher who's walking along it and collecting information. So basically that is what it is. And then we have aerial photographs. So sorting through quadrats by hand is often too labor intensive. So if you can think back to those previous pictures and that small amount of space that they were looking and finding, like that was packed full of plants and who knows what we couldn't even see, that's pretty time intensive to be able to spend the time and the labor to go through those throughout an area and really get a lot of information. So they really don't translate that information very well to a large area for biodiversity. Instead, this is where aerial photographs are often used. So there are common types of film used in aerial photography, but it could be black and white, infrared, or color. So it's not like you just have one way to do it. Depending on what you're looking for, you might use different types of films to take your pictures. So black and white and color hopefully are fairly self-explanatory, but infrared is on the electromagnetic spectrum. So you think back to when we were talking about radiation, and we were talking about ultraviolet light and how that's on the one end of the electromagnetic spectrum outside of visual light. Well, infrared is on the other end. So ultraviolet are really, really small wavelengths, and infrared are much, much longer wavelengths. So it lies beyond red at the end of the visible light spectrum. So that means that with infrared film, researchers can record and see what the human eye cannot see, because we can't see infrared. So if we take pictures, that means we can see information that we can't see otherwise. So here is 3 images of the same space that are in different, have different filters or different films on types. So we have color. Also, the roads have been drawn on with digitally, so those are not naturally in the photograph, but they've been sketched on. So we can see that we've got green trees, we have some sort of settlement or something going on here. Then we have black and white, which shows us different contrast. Different things pop out in different ways. Then we have infrared, where now we can see, again, our information shows up in very different ways. The trees are not actually red, but they're actually green, but they show up as red based on their reflection of infrared light. I don't honestly know why you might use one or the other. It's not something that we need to know. We're just, at this point, we're just understanding that there are various options. So Canada has 5.5 million square kilometers of forests and other wooded lands. So area photography helps researchers monitor the biodiversity of these forests. It is very, very difficult for people to be able to measure or look through all of those areas in 5.5 million square kilometers of forest. That's a lot of wooded area. So be able to take pictures and gather information is really, really useful. So for example, Canada's National Forest Inventory uses aerial photos to make estimates about species biodiversity of trees, as well as gather data about forest health, so like insect damage or disease, and productivity. So using ground sampling and area photography together, they measure a network of sampling points across Canada at regular intervals. By collecting data over time, they can determine the state of Canada's forests. So now we're going to look at the Lincoln index and Capture-Mark-Recapture sampling, specifically that goes along with this, the Lincoln index. So this is used for sampling animals, birds, and fish, because plants and trees are really great at basically just staying where you found them. So they're easy to come back to, they're easy to get a bigger picture because they are not moving. But to be able to sample animals, birds, and fish, this is much more difficult as they are on the move. They don't generally want to be caught. They don't want to be tracked or marked or counted or researched upon. So they don't volunteer that. So this is one method to be able to collect data about moving animals. So it's used to estimate the number of individuals in a population of mobile organisms. So the fact that they are moving organisms, and that we're estimating how many they are without actually counting each and every one. The index is based on Capture-Mark-Recapture sampling. So how this works is that a sample of organisms is first captured using various methods, which we'll talk about. And then we're going to talk about how they're captured first. So first, we capture some animals. So there's different ways depending on what it is that you are capturing. The first one is the aquatic dip net, and this is used to collect aquatic invertebrates in shallow water. So it's swept through the water or pushed through aquatic plants to collect insects and bottom-dwelling organisms like worms and clams. So we're not catching fish, but we're collecting other grubby things from the water. So hopefully my links will be working and we'll be able to see a little bit of a video of how this works. No, this isn't.
So I've got my essay written, and I've been working on it for about
just
a week.
an ad. We
So now I'm going to show you how I use randomly.
There we go.
You will collect one dipnet sample per site. Each dipnet sample consists of 2 dip netting efforts. Dipnet in the near shore vegetated zone, in water up to 1 meter deep. Sample close to the edge and into the vegetation. Avoid sampling near the bottle traps if possible. Before you take your first dip netting effort, put water in your collecting pans that sit underneath your screen. Place the screen over the trays so that its edges don't hang over the edge of the pans. This way, the critters go down into the water in the pans. Sample in the shallow emergent vegetation zone in water not deeper than 3 feet. Hold the long-handled dip net vertically, one hand near the net and one hand up on the handle, much like you would hold a canoe paddle. Using strong strokes, sweep the net through the water towards you about 3 to 5 times. Be sure to sample into the vegetation near shore. Dump all of the net contents onto the hardware cloth screen. Get everything out of the net. Spread the vegetation and loosen it. Do this periodically for up to 10 minutes. This allows the critters to get down into the water in the pans.
10 minutes. Okay, great.
After 10 minutes, remove the vegetation.
Done?
You are now ready for the second dip netting effort. Move to a different shallow area and repeat the process. After spreading out the vegetation for 10 minutes, remove it and pour the water from the collecting pans through your sieve. Be sure to pour the contents over the large pan in case of spilling. Dislodge the leeches and snails that might attach to the pan. Backflush the sieve contents into your sample jar with 95% alcohol. Be sure to get any critters that are attached to the sieve walls. Use 2 jars if critters and debris occupy more than 1/3 of the jar volume. Once your sample and alcohol have been placed into the jar, be sure to label the jar properly. With a pencil or India ink, write the site name, date, type of sample, and number of jars on a heavy piece of cardstock or note card. Place the label inside the jar, then label the outside with tape.
All right, so that shows us how how dip nets work. So it's really, you're just putting in, here's a pretty good picture of the net. You go into the water and you're grabbing bugs and worms and slugs and leeches and things like that to then count and test and track them. Then mist nets are the second type of capturing, depending on what you're capturing. So this is a fine net attached to poles that are set up and stretched out to block bird or bat flight paths. So this is how you can catch some birds, probably not all the birds, but some birds. The nets are so fine, so it's difficult for birds or bats to see them. Then once caught, the birds or bat falls into the net's loose flaps and are quickly removed by researchers. So they don't stay in. So here's a picture that's kind of close up. They don't stay in these nets. The nets are just, the birds get caught in them and then the researchers come and get them out really quickly. So let's see if this one will work for us.
The following banding techniques are used for our tidal marsh sparrow project and can be used with other songbird species. We use 12-meter long nets called mist nets to capture these sparrows. The mist nets are 2.5 meters tall and are suspended between 2 poles. Since we are in a salt marsh with low vegetation and no trees, we can prop up poles in the soft ground with the appropriate support and tension without using ropes, stakes, or trees to anchor the poles. A very important step in setting up mist nets is making sure the area is clear of any vegetation which can injure birds caught in the net. Since we are in a salt marsh, this is often not an issue. Once the nets are properly set up, we return to our banding station and wait for the birds to fly into the nets. Since the nets are thin and thread-like, it looks invisible, and therefore birds will passively fly into the nets because they cannot see them. Since the nets are often in direct sunlight, we check them at least every 15 minutes. We check every net and make sure that all birds are extracted and that vegetation or insects are cleared out of the nets before returning back to the banding station for the next round of net checks. Our banding station has all the tools needed to process a bird. These tools include calipers, bands, banding pliers, wing cord ruler, and various buffers for our fecal and blood samples. We will also actively drive birds into the nets by running and clapping. We aren't always successful in getting a bird into the net. We corralled this bird into the net. Even when we don't get a bird, we always have to check the nets. Here we successfully caught our female. Unfortunately, playback, which is the use of...
So we're not going to go into more detail. The video goes into more. If you're interested, you can check it out. I'll put it in the show notes and link it in the supplementary videos. But you can just see how the nets kind of work. It's a little bit tricky to see just because they're so fine. That is kind of the whole point. And they obviously in respect of the birds and not to stress them out. They didn't video them really close while they're in it. But now you can kind of see how that works and how these birds are in it in this close-up picture. Then we have helicopter net gunning and darting. So this is often used for larger animals like wolves or grizzly bears. Using helicopter nets are dropped over fleeing animals. Then animals are shot with tranquilizer darts to temporarily immobilize them. Again, I didn't find a good video where you could see them actually shooting a net from a helicopter, and then the ones that I could find just seemed more like hunting. I didn't like the ones that I found. So here you can see that someone is shooting a net from a helicopter onto a deer or something to then immobilize them so that they can then check them out. Here is
cutting-edge casino technology. Leo Vegas.
ad.
Jana from Wild Scars Aviation. They do our chopper flying for us and our...
So this is one, so they're doing some rhinos. So I'm gonna start up from where I wanted. So we're just gonna jump, here we go, back to the helicopters. So you can sort of see, I don't know why I won't, it doesn't like me.
Jana brought in the research program. Got it, brought it Jana from a helicopter. You have to get in really low, you have to get in really close. So the competency of the pilot is absolutely critical. It's a vital part of our program. Jana is an amazing pilot, knows how to get the job done. That's just a really nice operation and nice people to be able to work with.
The team is ready to go and communication just came in from the helicopter of the rhino's whereabouts. It's all systems go. I got the camera set and on record, and Kevin drove the team to the location where we had a visual on the chopper, and then we knew the rhinos were close by. Dr. Peter Rogers is using an Excalibur CO2 darting rifle, which shoots a dart at 350 feet per second. This allows the projectile to penetrate the rhino's thick hide and deliver 80 milligrams of butorphanol into the rhino. This is enough medicine to knock out 160 men. The vet wastes no time and makes a perfect shot. Jana, the pilot, radios in and tells us to start our stopwatches, saying the darting of the rhino was a great success. She skillfully guided the rhino with the helicopter towards the road where we were waiting, and eventually the rhino went down within 30 meters of the vehicles.
Now, once the rhino goes down, a lot needs to happen. Everybody's got their own job to do, and everybody knows what to do. Rhino...
So then they continue to go and show how they then treat this rhino in terms of tracking it and taking it and taking some samples. But the helicopter and the dart gun is sort of the key in terms of how they are able to get close enough to this large animal, rhinoceros. And then finally we have live traps. So there are many different kinds of live traps, and they're focused on capturing organisms in as human a way as possible. So are closely monitored to prevent organisms from remaining in them too long. So similar to the mist nets, they don't want to trap animals and have them being stressed out and hurt themselves. Examples include baited foot hole traps with rubber padded jaws, cage traps, and camera traps used to study shy or nocturnal, which are active in night organisms. So cameras are connected to infrared beams that trigger whenever something breaks the beam. So then that's one way you can study nocturnal animals. So I have a quick video of turkeys being caught in a live trap. Oh, come on, just work. Yeah, all the other ones worked. Sorry, it's entertaining. The turkeys are fun. I want you to see the turkeys. Let's see if I can. Oh, again. Okay, so we don't need to watch them set up the the trap. This is totally not my video. We don't want hunting.
Our primary focus is to look at the habitat use of Rio Grande wild turkeys in South Texas. There's a lot that goes into it, but it only takes just a couple seconds to happen.
In go the turkeys.
When the net goes off, our primary goal is always the safety of the bird.
There goes the net.
Drop. It's pretty exciting when that net drops. Hey, bird's on the edge! Back corner! It is very fast and furious. We're not underneath if we need to. We try to do it as fast as we can and reduce stress on the birds. Who do you guys saw? It's quite a rodeo sometimes.
All right, so you can see how the live trap works to get the turkeys. So there's the various ways that animals are captured. So then the organisms are marked and released back to mingle with their populations. So once they've been captured, they get tagged in some way. And then later, so this could be hours, weeks, months later, depending on it, they capture a second sample, probably the same way they captured the first. And then individuals from the second sample that are not marked, so none of them are marked, but they calculate the ratio between the marked and the unmarked organisms in order to estimate the total number that they have altogether. So this is done through this population, this formula where it's the total population is equal to the number of organisms captured and then marked in the first sample times the number of organisms captured in the second sample, and then all divided by the number of marked organisms in the second sample, and that gives you an average of how many animals there are in total. So the following conditions must exist for this sampling to work. The population of the organisms must be closed, so there can't be any things that are coming in and out of that group. The time span between taking the different samples must be short compared to the lifespan of the organisms. And the marked organisms must mix completely with the rest of the population in order to really know that you're getting good information from the whole sample. So that's how we sample them, but then how we track and tag them. So sometimes organisms are simply marked for researchers for possible future identification to just see how they're doing and what's going on. So migratory birds are one example, and they're tagged so that their migratory patterns and survival rates can be collected and compared in various parts of the world. They can use alphanumeric labels, so this could be a small metal or colored plastic ring or flag, which is stamped with a combination of letters and numbers and then recorded. So here are some examples of flags on birds' legs that have numbers on them, or here even in a fish, it's an implant that goes under the skin so that they can track that fish. Then there's also wildlife radio tracking. So this one transmitter collar that uses radio signals is tuned to different frequencies that can report individual animals' movement, behaviors, and as well as the population size of their community. So here are some images of various types of animals that have these tracking on them. So you can see these collars on these various different animals, which seems very strange to me, these wild animals that have these trackers on them. These collars on them. I don't know how I feel about it, but anyway. And then here you can see a person using a transmitter to get information. And then the last thing, the way that they measure track animals is from space. So researchers also use Earth observation, which could be EO stands for technologies, to measure and monitor biodiversity. So EO technologies include individual satellites and global positioning systems, so GPS, just like we use in our cars and on our phones, that are able to collect and computerize images and data from space. So GPS technology involves a system of satellites and portable receivers that are able to pinpoint a receiver's location anywhere on the Earth's surface. All right, so now you can do the key questions on page 167, questions 1 and 2. All right, so that is all of our information about measuring and monitoring species biodiversity. So this is wrapping up chapter 6. This is lesson 20. So we talked about sampling. We talked about the different ways of collecting information, either using Quadrat, line transect, or aerial photography in terms of sampling stationary plants. Then we talked about the Lincoln Index, which used the capture and the Mark-Recapture method of collecting information. and we looked at aquatic dip nets, mist nets, helicopter net gunning and darting, and live traps for ways of capturing the animals, and then the different tagging ways that you could use to then, which is next, various tagging ways, and then releasing them and capturing them again. And then we talked about the different wildlife tagging tracking. So we talked about the alphanumeric flags and rings, the radio tracking transmitters on collars, and then also the GPS tracking systems, which is using satellites in space. Alright, so hopefully you can explain the differences between quadrat, line transect, and aerial photography sampling. You can also explain and calculate the Lincoln Index. Sorry, I rushed through that a little bit. I was worried about time, that we weren't going to get through everything. But remember, that is just where to find out the total population, to estimate the total population, you take the number of animals that from your first sample that you marked, so the total number of animals that you marked in your first sample, multiply it by the total number of animals that you've captured in your second sample, and then divide that by the total number of marked animals in your second sample. And that will give you an estimation of the total number of animals in that population overall, because it's like you're doing a ratio. You're saying you're comparing the number of animals that you sampled, that you marked, compared to the total number of animals that were marked within your second sample. If you have a hard time with that, let me know. Feel free to reach out to me and I can walk you through it a little bit better, the math of it. And then we talked about the different tracking methods. Hopefully you can describe the alphanumeric, wildlife radio tracking, and GPS methods of tagging and tracking. So that is, then you can do the questions on 169, questions 1 through 13, where you can see you have to do a little bit of math, not a lot, a little bit of math, but then also continuing just our regular questions about explaining the differences and the similarities of things. So that's a wrap up of Lesson 20, Chapter 6 again. So we've finished Chapters 1 through 6, and we have Lessons 21, 22, and 23 to conclude this week, which are Chapter 7. So we'll be looking at a different topic than we've been talking about before. And then we will be finished, and then you'll hand in all of that work, so submit all of your work by June 10th, but also earlier, so that you can also do your commenting assignment that you'll need to do before June 10th as well. If you have any questions or concerns about anything, if you're struggling with something, if you're not sure if you can get it done and you want to talk about options, please reach out to me. My number is 807-737-1488, extension 2209. You can also call us toll free at 1-800-667-3703. You can email me at bronwyn.slate, and that's spelled BRONWYN.SLATE@nnec.on.ca. You can connect with me through Facebook at bslate Wahsa, and you can watch all of our lessons on YouTube, which is also called bslate Wahsa. I'm here at the office Monday through Friday, 9 a.m. to 4 p.m. We are not open to the public yet. Well, that won't happen this year, maybe hopefully next year, but if you want to reach out to me, that's the best time for me to either catch me on the phone, or I will get back to you really quickly if you email me or Facebook me, generally, unless I am teaching or in another meeting. So feel free to do that. Yeah, thank you so much for joining me. I hope this was helpful. Enjoy your day, Witch.